Alrighty, this is a little bit of a different format, but work with me here. So I'm going to be talking about the composition of blood, and then how the blood coagulates, and the platelet plug, and then the cascade, the coagulation cascade. Yeah, and that'll pretty much be the whole video. So first, I want to talk about what the composition of blood is. So if we were to have a vial, and we were to separate the different parts of blood, we would have three different looking substances. We would have red blood cells. We would have a buffer layer. And then in the buffer layer, that's where you find your white blood cells and antibodies and stuff. And then we would find the rest of this. It's kind of like, I think it's a yellow color. This would be plasma. So if we were to make a chart for plasma, it is based off of charge. I'm not sure like if this has a name or if it's like called something, but it has a charge. So if you were to separate all the things found in plasma into charges on a graph, you would find a couple distinct sections. You have a parabola, a bump that represents albumin, which is a protein found in your blood. You would have an alpha and a beta bump, and then you would have a gamma, and then the gamma is where your immunoglobulins are found, which those are all your antibodies. So how your body fights infection and fights disease and stuff are found here. So when you donate plasma, or when someone receives plasma, it's usually in order to receive antibodies for obviously something that they don't have. And so a red blood cell has a biconcave shape, and so this would be like looking at a red blood cell from the side. Red blood cell. So inside this guy is something called hemoglobin. So hemoglobin is 4 different subunits of just proteins that are intertwined together. And then in the middle of each of these proteins is found something we call a heme. And a heme is 1 iron. So 1 heme equals an iron connected to an oxygen molecule. And so for every 1 hemoglobin, you have 4 subunits which have 4 hemes, which can carry each 1 oxygen. So 1 hemoglobin equals 4 oxygen carried in the blood. But red blood cells don't live super long. They only live like 120 days. And so how your body digests this, I guess, is through macrophages. Macrophages. And these guys eat red blood cells, and this happens in the spleen. It filters out all like the damage and all of the old red blood cells, and so each of your little hemes kind of looks like this fun little pentagon star thing, and then in the middle is where your iron is found. So this is a heme. Here's your iron, and then we have little nitrogen molecules over here. Nitrogen. And then through bacteria, this transforms from a heme to something called we call biliverdin, and then another very similar looking structure called bilirubin. And bilirubin is what's going to be digested by other parts of your body. So it looks pretty similar, other than right here there's 2 double bonds to oxygen, and then there's nothing in the middle, and then hydrogen and nitrogen are found in these little subgroups. Nitrogen, hydrogen, nitrogen. So this guy goes to your liver. And this guy takes bilirubin and it adds glucose or adds different sugars onto it. And that is what we call, it has become a conjugated bilirubin. And so through your bile duct or bile, it enters into your intestinal tract. And so this conjugated bilirubin is going to turn into UB, which is urobilirubinogen, and then stercobilin. Stercobilin is going to stay and be further digested in the intestinal tract, and this is brown in color. UB is going to, through the bloodstream, is going to go to your kidneys, however, and be turned into urobilin. And urobilin is yellow in color, or like light yellow in color. So this is what your blood is made up of, and then how red blood cells are like digested, I guess. Okay, and the next thing I wanted to talk about was just how your body clots blood, because that's super important for like any wound care, anything that happens to your cells, it needs to be able to clot and like plug that hole. And so we have, I'm going to present this as a blood vessel. So there are all these little epithelial cells on the inside of this blood vessel, and then surrounding this blood vessel is what we call the basement membrane. And so if we were to have a tear in the blood vessel, it would expose the basement membrane here, which is important. And then the basement membrane also has all these little negative charges on it. It's negatively charged. So inside of the bloodstream, we find platelets. I'm just going to draw like little squigglies for platelets. And then something we call VWF. And then on the platelets, we have GP1B, or those are VWF receptors. And so these guys are going to combine together. They're going to attach to one another. And then the VWF is very attracted to this negative charge in the basement membrane. And so when VWF binds over here with a platelet, blood is still flowing, which activates mechanoreceptors on the platelet, which activates it. So this guy spreads out its arms and goes, woo, woo. And then this activated platelet does two things. It will release ADP, and then it will also release something called thromboxane, or TXA2. And both of these guys are going to activate other platelets, because we have TXA2 receptors as well as ADP receptors on all of our little platelets. And so when one is activated, a bunch of other platelets are also going to be activated. So I'm going to draw another picture for that. So we have this, we have the little hole, we have this guy, we have our basement membrane, and then we have our activated platelet. So this guy is going to be surrounded by other activated platelets because of what we released and then how their receptors were affected. And then all over these activated platelets are going to be found something called P2Y12 receptors, or fibrinogen receptors. Something else to know about activated platelets is they have dense granules, and they have alpha granules. So dense granules is going to be the ADP that's released, and then that's pretty much the most important thing. But another thing to mention is that the alpha also releases fibrinogen. So these fibrinogen are I'm going to represent them as little coils. They're going to coil these platelets together, which that's how they're attaching, and that's how they're creating the platelet plug here, and because of those fibrinogen receptors. Also happening at this active site is something we call a cascade. So outside here, there's obviously the basement membrane, but there's also like cells, like there's muscle cells, there's adipose tissue, there's all these different there's mast cells, and all of these guys are releasing something called TF, or tissue factors. And these tissue factors, or this is also called factor 3, these tissue factors are going to affect factor 12. So when factor 12 attaches, it's going to turn into factor 12A. Factor 12A is then going to affect factor 11, and then that guy is going to affect factor 9, which is X1, X, and then this guy is going to affect factor 10, and then factor 9 also needs factor 8 to work, plus factor 8, and then factor 10 also needs factor 5 in order to work. So factor 10 affects factor 2, which is called, factor 2 doesn't have one, but an activated factor 2 is called thrombin. And then thrombin is going to activate factor 1, and then factor 1 which is called fibrinogen. And then that is going to turn into factor 1A, which is fibrin strands. So anything before factor 10, I know this is kind of a wonky picture, but anything before factor 10, this is what we call our intrinsic pathway. Because we have another pathway that is kind of like a fail-safe. They work together to both clot blood, but it's important that we have two of them so we're able to control that, or if one pathway fails, then the other one will continue working. So we have these, the basement membrane, we have these different cells out here releasing TF as well as factor 3, but we have factor 7 that comes in here and gets activated, and factor 7 can also activate factor 10. And so then this goes back to the common, this is called the common pathway. So because this is not dealing with inside the blood, this is going to be called our extrinsic pathway. So a couple of drugs that affect both the intrinsic, well, not at the same time, but affect the intrinsic pathway as well as the extrinsic pathway, is the intrinsic pathway can be affected, I'm just going to represent it as a big I, is going to be affected by the drug heparin. This is because it's going to affect factor 12, so then it can't cause the cascade. However, if heparin is something that we just naturally release, but it's something that you can also take, like, just as a medication. However, if we affect the intrinsic pathway, this extrinsic pathway is still up and working because it has to do with factor 7, not factor 12. So there is still clotting in the blood, it's just less effective. And so if someone only had the extrinsic pathway working, eventually their blood would clot. They will probably die of hemorrhaging first, but like their blood still clots. Like, it's not a super big problem, kind of. But to test for intrinsic pathway, you do a PTT test. And then for the extrinsic pathway, which I'm going to represent with a big E, you would give something called warfarin or Coumadin. This is going to inhibit vitamin K, because vitamin K helps produce factor 7. So if we inhibit vitamin K, then we're not making any factor 7, which stops the extrinsic pathway. However, if we give someone warfarin, their extrinsic pathway isn't working, but their intrinsic pathway is working. And so one of them is always working at this. In an ideal situation, both of them will be working, but hopefully we can keep one working. And so a test to look at the extrinsic pathway would just be the PT test. Yeah, and that's all about blood and how it clots.